So good morning, everybody. Thank you to Rose Trees. Thank you very much, Steve, and thank you to the organizers for the kind invitation to speak this morning. I decided to give quite a general talk. So I spend most of my time developing new antibiotics. So connecting some of the basic science that I've seen in the program to phase 2 and 3 clinical studies. I occupy that space. But the talk I'm going to give doesn't really focus so much on that area. And I just want to really try and hone in on opportunities, I think, for quantitative sciences to really fix significant problems in this space. So first of all, my comments today are about human health. And I sort of don't mean in that sense to upset anybody by not acknowledging the importance of environmental health and animal health in this space. So AMR is one of the major problems of our times, but all I really know is human health. So, and as I said in the introduction, my talk's general, and I'm looking for those opportunities for us to work together across disciplines to solve problems that haven't been addressed for the last 20, 30, 40, and 50 years and continue to cause trouble for us. So I just want to take some time now to really tell you what I do for a job, but also to point out an important point of difference about where I think the AMR field is and some of the words that we use. So here's what most people mean by AMR. This is what gets onto Radio 4 or in The Guardian or The Times. And here's a bacterium that's come from a transplant patient. And then on the left, is an antibiogram, and R stands for resistance. This is what a clinician would see when trying to select a drug to use for this patient. And we are seeing these organisms increasingly in the world, but certainly in UK hospitals where there's maybe one or sometimes no active drugs that can be used. And this obviously causes a lot of anxiety, and this sort of drives the development of new drugs. And this is what people mean by superbugs, multi and extremely drug resistance. And this is what most people think is the AMR problem. But for me, it's not really the only part of the AMR problem, which I think is much bigger. So you saw in the title, the inverted U, and why is this important, and actually what is it? Well, I think that the inverted U helps explain the boom-bust cycles of antibiotics for the last 50 years. So the last 50 years has been figuring out how to make antibiotics, exploiting novel targets that, I've seen in this program, making drugs, and then those drugs being eroded with time and becoming less useful or sometimes completely useless. And so then what we do is we pick a new target, we make a new drug, and we go around this cycle again. And that is actually, I think, a failing model and something we need to address. And I guess that's what a large amount of this talk is about. So when you this is a hollow fiber model, which we have in Liverpool. Hollow fiber models use a dialysis catheter, and their advantage is that you can simulate clinically relevant concentration time profiles of drugs. So many early experiments are done in the lab with fixed concentrations of drug. Here you have concentration time profiles that are human-like. And just to look at this slide, just to take you through this slide. So the red open triangles there are the real data points, and the solid red line is the fitting. And you can see these are untreated controls up there on the top left. Tebipenem is a carbapenem, so it doesn't really matter what tebipenem is. The blue line is a resistant subpopulation that's able to grow on drug-containing agar at a certain threshold. And you can see in the controls, there's no expansion of that resistant subpopulation. But as soon as you apply drug, and you can see the drug being applied there on the right in the dotted black line, you can see that's the concentration time profile that's relevant to humans that's being used in the experiment. You get some decrement of the bacterial density in the red, but you get this rapid expansion of a resistant subpopulation. And that is seen pretty much consistently for many drug pathogen combinations, and is also seen in the clinic. So what is this? So what this is this is not what gets reported in the Guardian or on Radio 4 as a superbug with fixed hereditable genetic changes. These are a series of dynamic mechanisms that come into play as soon as an antibiotic is applied. So this might be, for example, downregulation of porins. So the antibiotic has less access to its site of action or activity. Or it might be upregulation of pumps, so that the drug is extruded. Or it might be an interaction of these mechanisms. But whatever, these are the mechanisms that preserve life and result in phenotypes that are able to resist the antibiotic. So they're not stable in the sense that if you take the drug away, then there's reversion. But this is the earliest forms of drug resistance. And so when you, and this is what I really do for a job now, is that I map out these relationships. And this, I'm sorry for this slide being slightly busy. Maybe we should just concentrate on the blue line. So in the top left, there are the controls. You put a bit more drug in and you get this emergence of a resistant subpopulation, a bit more drug, more emergence of resistance, and the same in D. But as you keep scaling the drug up, winding the drug up, then that resistant subpopulation disappears. And when you look at the last time point, as it were, and you show all of those panels as one, here it is. Here's the inverted U. So when there's no drug pressure, you have a resistant subpopulation, but that they are few. When you have a bit more drug exposure, you get this maximum expression of mechanisms that preserves life, as it were, enables its emergence of resistant clones. And then as you scale up higher, maybe to beyond what's clinically feasible, you switch everything off. And there's the inverted U. And the inverted U was really first, it sounds so simple when you say it, right? But it was George Strasano, who's my sort of friend and mentor, that really described this. And so here is the problem of AMR in a single slide for me. So the red is effect, right? So that this is for any drug, you have the red line, which as you increase drug exposure, you get more and more effect. You can't keep going forever because you get the emergence of toxicity. That's in the black line. But for antibiotics, you get this inverted U right in the middle of those two curves, right where you'd like to put the exposures for a drug. You have this thing called the inverted U. So antibiotics actually have three relationships, and that's what makes them different from many other therapeutic agents. And so to successfully manage the patient to make them better, you have to manage the red line and the black line. But you'll pay a price every time you try and do that, because you'll get the emergence of resistant clones. Now, what happens, of course, depends on the clinical context. Whether the patient's going to be in hospital for long enough, the density of organisms, whether they transmit, all of those things are a little downstream. But the point is, as soon as the drug's applied, this relationship is generally in play. And you can also see another thing, just to say, is it's almost impossible to design a regimen that simultaneously resolves these three curves. You can't put it in a place where you have maximum effect, minimum toxicity, and you don't get any emergence of drug resistance. So that, I think that's really the problem. And so if you do accept that that's the problem, I'd be interested to see whether you do, what can we do about it? So what can be done? Okay, so the inverted U, just to say it all once more, results from the evolutionary machinery that preserves life. It's operational every time an antibiotic is used. And so here is AMR, therefore, is intrinsic to all modern antimicrobial chemotherapy. There isn't infection without AMR versus infection with AMR. So I hear this debate all of the time, and it drives me crazy. So infection is different from AMR. Well, I'm sorry, it's not. It's all one and the same thing for me. And then again, just to summarize here, the early changes that you see that are happening right now in the NHS for so-called sensitive organisms or for wild types, you have these early epigenetic resistance mechanisms. They're dynamic, they're fully stochastic, which makes their observation in clinical studies difficult. You do the experiment and then you try and reproduce it, and something else happens and you think, what am I doing wrong? But these are stochastic mechanisms. They interact. they ultimately enable survival in the short term that leads to hereditable genetic changes downstream, but it's late. And so that's on the other side of the spectrum is what gets reported in the media. These are fixed, hereditable, stable mechanisms that spread around the world. So MECA for MRSA, ESBLs that are endemic in UK hospitals, KPCs, endemic now in some parts of the UK. These are sort of, they behave more deterministically so that if you have the thing and you apply the drug, it fails. But it's generally what people mean by AMR and superbugs. And so it's a distinction that is maybe subtle, but important, I think. So what are the problems that need the help of physics, chemistry, engineering, mathematics, computer science? I don't know whether I get across all of the things that you mean by quantitative sciences, but how can they be applied to manage the inverted U? So I was chairing after I stopped working on COVID full-time, like many people on this screen, in the middle of last year, and I was chairing a meeting of a new antibiotic, and I sort of got really depressed, and in the sense that I'd been through all the learnings of COVID, what a phenomenal response of biomedical science that this country and the rest of the world showed in tackling some of those issues. And I just felt that the AMR debate hadn't moved on for all of that time. Okay, there might have been reasons for that, but the thinking hadn't moved on. And so I sort of fired off on a Saturday afternoon a series of tweets, which actually caught some eyes, and I've put some of those in the talk here, just to say these are the things that I think that people we don't pay enough attention to in sort of being creative. We're sort of stuck with a paradigm that's not working. The first is diagnostic and deployment of antibiotics. So we do know that early appropriate antimicrobial therapy saves lives. There's clear data to show that. But this, on the other side of the coin, leads to gross indiscriminate use. And that's facilitated actually by the safety of antibiotics. They are incredibly safe things to use. They're low cost. This is true. The tools and approaches and thinking to deploy antimicrobial therapy haven't changed in my working life, which is now approaching 3 or 4 decades. And it's just, we just keep going around, and what's the definition of doing the same thing and expecting a different result and all of that. And so, and then this is the other thing that drives me crazy. I'm not a diagnostics guy, because I can say this. So everyone says we need new diagnostics, and that's part of Jim Lord O'Neill's 10-point action plan. So here's the first opportunity for us together. So most ideas of diagnosis and diagnostic technologies, of course, have historical precedent. They can be traced to the work of Koch in the 1870s and 1880s, when he isolated anthrax and tuberculosis. He discovered agar, showed that the causative pathogen could be isolated outside the host and it could reestablish disease, and you get Koch's postulates. And that basically has underpinned everything that still happens right here to this day. So most clinical care in the UK is directed on the basis of isolating the causative pathogen from the patient, and blood cultures are especially relevant. So, that's part of my job, and others' job is to monitor the blood cultures coming out of a hospital and reacting to positive blood cultures. So one thing that's failed with that is that molecular testing hasn't been as disruptive as was certainly the case for viruses like SARS-CoV-2. This is a molecular disease, but that hasn't worked for bacteria. The other thing to say is the sensitivity of blood cultures is extremely low. I mean, for some diseases, 50% might be optimistic, actually. So this leads to the first problem, and that most antimicrobial therapy is empirical. And I'm not that sure that people outside the clinical arena know that that's the case. And actually, my job as a clinical doctor is easy if you have the organism in hand. You actually generally know what to do. But most therapy is empirical. You have no idea what you're doing. And so I just wonder, this is the first chance for us then, maybe. So that here are the opportunities maybe for disruptive thinking that require the physical sciences and quantitative techniques. So real-time testing at the bedside, inline monitoring for infection, detection of things that are not living, so metabolites, volatiles, proteins, and there's great work in this area. But I think that the real opportunity might be with machine learning and AI to escape this current microbiological paradigm. So in other words, the decision about whether to start or withhold therapy has to be made without laboratory data. I think that model seems to be broken and out of road to me. And so also the development of risk-based algorithms to guide appropriate antimicrobial choices. That would be another important step to make. And then there's this other problem of assessing clinical response. So that is done very crudely at the moment. So using computers and algorithms to decide when it's safe to stop antibiotics, or when, on the other hand, when it might be important to intensify therapy. Okay, grand challenge number 2. So we're completely obsessed with a one-size-fits-all, one regimen, one breakpoint, one duration, with very little interest in biological variability, outliers, and special populations who drive the emergence of drug resistance. So the healthcare service needs to care for everybody, not just the average patient. Okay, well, so I think everybody will say, yeah, well, sure, that's fine. We agree with that. But actually, when you stop and think about it, this is completely counter to all notions of engineering. I mean, my favorite example is that you would be very sort of nervous if a 777 was flying across the Atlantic with a one-size-fits-all approach. So engineers don't accept actually one-size-fits-all. Culturally, they don't accept that. Much of what they do is accepting that there's uncertainty and reacting and managing that uncertainty. But clinicians, on the other hand, just double down and say, I'm sorry, one size fits all. And this is a cultural problem that needs to be overcome. So, and it's real. I mean, so look at this. So this is the concentration of a drug that's used extensively in the NHS. This is voriconazole. This is actually the concentration time profiles given to healthy volunteers. And you can see there's absolutely extraordinary variability in the drug exposures of between patients. And you remember, this is all, in a sense, unpredictable. There's no way that you can figure out why one patient's doing one thing and why another patient's doing the other thing. And then the 2 red lines there are showing what might be considered a safe and effective range of concentrations. And so you can see a lot of patients fall outside that range with a one-size-fits-all regimen. So this is an engineering feedback loop, and here it's been adapted for therapeutic drug monitoring. Systems the patient, outcome of the drug is making a patient better. Measuring, well, it might be a drug concentration, a target, and then the physician's the controller. I guess my point is that these systems need to now be employed as routine. Now, we've done some of this in Liverpool. This was actually a real world example of voriconazole again, where we learned in the first 2 dosages that I'm showing you there, and we made some software where we could individualize dosing to bring patients to where we wanted to bring them back into what was safe and effective concentration. So I think that this took an awful lot of work, and you know, it didn't really raise a blip on anyone's radar. But I think that this is the work for the next 10, 20, 30, 40 years, making this much easier to do and much more mainstream, and getting away from the one size fits all, which is basically, that's a problem of the quantitative sciences. And so here are some examples. I'm going to actually skip over that, Steve. The example of inline monitoring. So part of the problem that we have is that measurement just takes too long to make changes in regimens. And so this is work from Alison Holmes' group at Imperial, where using real-time inline monitoring of drug concentrations as detection mechanisms, and then algorithms that can be designed then to adjust dosages and take patients back to where you want them to be. So the third one for you is one that's really worrying me at the moment, and that we have no way of generalizing findings from sanitized clinical trials, which were necessarily restrictive to create homogeneous populations that enable a drug to be initially licensed. So there's this massive step from a licensing study where maybe 1,000 patients or so might be studied, to when that drug is unleashed onto the NHS population, where hundreds and thousands of patients may receive a drug. It's essentially an unsupervised step, And there are some problems with that, I think. So I think that we need system-wide approaches that manage antibiotics as societal assets. And there's this large step, as I said, from small sanitized data sets up to these real-world data sets. And we lived through this just recently with the vaccines. Remember that the Pfizer and the AstraZeneca clinical studies were done, but then we started seeing the emergence of side effects or adverse events with the vaccines, but that only emerged at scale, and the ability to monitor and react to that was actually quite difficult for the health service because those data systems weren't in place. So the same actually happens with antibiotics. So we don't have good ways of reviewing their resistance liabilities. We don't have good ways of taking corrective action, and we don't have good ways of understanding how a system, and by that I mean a city or a country the size of the UK, will respond to the introduction of a new drug. And so then what happens is that we just use it, we get emergence of drug resistance, we lose the drug because of drug resistance, and the cycle goes around again. So again, how can physical and the quantitative sciences help? Well, sort of when I was depressed last January, sitting in these dark days of January, I think it was then, and I was watching the America's Cup from New Zealand, and I really liked those boats. But then I heard on the coverage about that whether to put a number 3 or a number 4 jib up or whatever with the boat, that was all done in simulators. So they would actually sail the race in simulators before they sailed the actual race. And I don't really understand why we don't do the same for new drugs in the health service for antibiotics. So there's this jumping problem, as I've said. And so I wonder whether there's an opportunity to actually try and find ways to simulate at scale, to decide how drugs should be deployed in the real world, to decide where the risks are and to mitigate the risks before actually doing the natural experiment. So here's the third sort of opportunity. So I'd be lying if I knew what agent-based models are, but you sort of know what agent-based models are. And so I wonder whether they can be developed for wards, for hospitals, for cities, for countries. And this enables these complex interaction of multiple things that might have an impact upon outcome. So it was actually physicists, and I think back to the Manhattan Project, that invented simulators because they didn't want to do the actual experiment of letting off a nuclear bomb. That had to be sorted in simulators. And so we make use of simulators in drug development, but they're not used, as far as I know, in the deployment of antimicrobials at scale. And then there's this idea of feedback control and corrective action and stressing systems, understanding what the implications are for understaffing ICU wards are, for example, in terms of the emergence of drug resistance. So Steve, I don't know how we are for time, but this is my last slide. So AMR, I think, is everybody's problem. So that's an important point, I think. It's just not a sort of a group of people that are interested in a very small subset of medicine. This is a profound biomedical and socio-political problem. The physical sciences and quantitative approaches actually have an absolutely fundamental role in solving these issues, and that I think requires multiple areas, multiple disciplines, and will require multiple creative steps. And I sort of look forward to those discussions with you. So here are my contact details, and with that, Steve, I'm done, and we'll stop sharing my screen. And
William, thank you very much indeed for an excellent and hard-edged overview of what life is like in this domain on the clinical front line. And there are a number of really big questions that you've posed as a challenge to the audience. Let me just put a few of the questions in the chat to you, and I'll just remind everybody that you'll have a chance to talk through in more detail William's excellent talk in the breakout room that William will be hosting at during the networking session. So Leonardo Mancini asks, I was curious to ask how the dynamic pulses of tebipenem are experimentally delivered. Is it just that the antibiotic degrades rapidly? I think that you had put in a sort of a series of a spike train, if you like, of the dosing.
Mm-hmm. Well, that just reflects, Steve, what happens in humans. So humans take the drug, it appears in the bloodstream, but then it is cleared by, depends on the drug, through the liver or the kidney. And so the pulse or the dose that you saw in the model system was simulating what happens or replicating what happens in humans. So we actually give tebipenem to the hollow fiber model, to the bit of plastic, and then we decay it away with pumps by replacing drug-containing media with drug-free media to simulate a first-order process. That's how that happens.
Can I just ask, in a sort of slightly linked vein, you beautifully described the inverted U, and clearly if antibiotics, there wasn't a problem with toxicity, the answer might be just to hammer it with the heaviest possible dose. But can you sort of sneak up on the high dose effect by holding the high levels of antibiotics, but for a very short time? And would that achieve greater lethality?
Yeah, so think that you get to the heart of the problem. So can you do that, and can you do that safely? And I think that actually, Steve, that there are times, I think part of the problem is it's just difficult with a single drug to do that. And this is the argument for combination chemotherapy. So to manage the U, you need more than one. Well, that's hardly a new concept, right? So that has been around since tuberculosis in the 1960s. But for more modern garden variety, if I can be so derogatory, antimicrobial chemotherapy, there are times when combinations of agents are required. But the problem then is that it's notoriously difficult to show that definitively in clinical studies. So then we say we don't have the evidence in clinical studies, we can't show a mortality endpoint. So then we retreat back to monotherapy. Then you get this emergence of drug resistance, but it's all delinked as it were. And then and around we go. So yeah, I think that, so the U, just to be, it does require insights into dosing, into precision, but then also getting out of the problem if you can't manage it.
Thank you. One final question. And as I think I mentioned, William, earlier, there are quite a number of modelers in the audience.
Yeah.
And Octavio asks, since the models require lots of data, how can this clinical data be made publicly available to the modeling community? I think that's a great question.
It is a great question. And it's actually, I've also, Steve, just recently been starting to think that this is bordering on unethical, what we do. So for drugs, so in Liverpool, my new thing now is to build a publicly accessible data repository for individual drugs. So, so for the geneticists get around, right? If you sequence something out, you put that in the public domain. Well, but for drugs, that information gets, doesn't happen. And it gets, stored on people's laptops, which then get left on trains. And then 30 years later, the data's not available, the raw data's not available. So I think that this is a fundamental point about data security, provenance, storage for public benefit, actually. And that is something we need to work on. And I think that also implicit in that question might be, we need to get a lot better at collecting data. So data, and I think SARS-CoV-2 has started to teach us some of the ways that can be done, so that these data are collected, but then stored for people to work on, and they're accessible for people to work on. Yep.